DNA, or deoxyribonucleic acid, is the genetic blueprint for life. DNA replicates through a complex process that was discovered by James Watson and Francis Crick. During DNA replication, DNA uses each half of a split strand to create a second or complementary strand, and the process is referred to as semi-conservative replication. DNA replicates during the S stage of interphase because it needs to occur before mitosis or meiosis. We must first understand DNA to understand DNA replication. DNA is composed of deoxyribose sugars, nitrogenous bases, and phosphates. The three of them combined is called a nucleotide. Sugars and phosphates connect to form two lines. Attached are bases, which are nitrogenous compounds. They attach to one another to connect the strands. There are four types of bases, all with different chemical formulas: thymine, guanine, adenine, and cytosine. Bases pair on either side of the helix because there is one base per sugar and phosphate. Adenine and thymine pair, and guanine and cytosine pair. Hydrogen bonds are used to connect the paired bases. The first step in the process is to use helicase, an enzyme, to split the double helix. Splitting of the DNA causes supercoiling, the additional unwanted coiling of DNA strands. Gyrase, another enzyme, works to uncoil the DNA mass. Each strand broken from the double helix is referred to as a parental strand because they create a template for their complementary strand. This is possible because bases can only pair with each other. DNA polymerase III, an enzyme also called an accessory protein, creates a complementary strand of DNA. Before DNA polymerase III can create a strand, a primer, a molecule that is the starter for another molecule, is required. The enzyme prionase uses RNA, ribonucleic acid, as a primer, making a few bases for the DNA polymerase III. DNA polymerase III fills in the rest of the complementary strands. However, one strand requires more primers than the other. One strand split from DNA is called the leading strand, and the other is called the lagging strand. The first is called the leading because it goes from a 5' to 3' order, meaning the first phosphate is attached to the fifth carbon molecule and the last phosphate is attached to the third carbon. The other strand is called the lagging strand because it goes from a 3' to 5' order. This is called anti-parallel. DNA polymerase III can only go in the order of 5' to 3', so on the lagging strand it runs backwards, requiring primase to put in more than one primer. This creates fragments called Okazaki fragments, which must be connected using covalent bonds by the enzyme ligase. It is also used in the leading strand. Even before ligase is used, DNA polymerase I, not III, replaces uracil with thymine. Similarly to how ligase is needed more in the lagging strand, DNA polymerase I is needed less in the leading strand. To complete the replication process, enzymes proofread the DNA strand. Without DNA replication, there would be no plants, animals, or bacteria on Earth.